So, good evening, everyone. Sorry, I was a bit late to start the class. So, we'll start the class directly. So, going on to the first question. So, we'll be doing discussion on week 5 and what we have studied in week 5 of this particular course. Okay. So, we'll be going into the first question. So, first question is poly D lysine is a what type of charge is present on poly D lysine at pH 7? Okay, so when we consider the structure of polylysine is like it's a common amino acid that we have. So it's the amino acid structure that we have for polylysine is basically it has carbon in the middle combined with hydrogen. Then on this has the carboxy terminal. This is the amino terminal that we have. And in the middle, we have CH3 groups, 4 in number, and followed by NH positive. Okay, so this is basically the common structure of lysine. And when it forms polyl lysine, then it forms a peptide bond with the other amino acid, other lysine, forming a peptide bond here. Okay, so now it will form lysine, 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 like this kind of structure. So it has a free amino group at some end, and at far end, it has a free carboxyl group. But it has one more group that is the amino terminal amino group that is its side chain. So it is also free in every case. So this amino group is free in every case. So now, what is the pKa value of this amino group? So the pKa value of this amino group is around 10. Okay, so for this question, we require the pKa value of particularly this amino group because this will determine the whole charge of the molecule. Okay, so we have a pH of 7, which is the basic pH at which the cells are basically cultured in our in vitro conditions. So like here the biochemistry comes. So what will happen if we have a pH of less than the pKa value? Okay, so what will happen here? The hydrogen ions are very high in this case. So these hydrogen ions, what will they do? They will go and bind to this N group, uh, combining H plus there. Okay, so that will form NH4 plus in that case. So it will give a positive charge to the molecule, to the whole molecule at pH 7. That's why it is used as a substrate so that it will already always have a positive charge on the surface and it will help in binding to the cells. Okay, so at pH 7, it will be a cationic poly because it is a positively charged molecule at pH 7. Okay, so it is a cationic poly. So the correct answer is option D. And here is like the explanation that I was explaining just now. So the amino terminal has the side chain has a pK of 10.53. So when we have a pH of less than like pH less than pKa, so in that condition, what will happen if we have high hydrogen ion concentration? It will go and bind to the cell. Okay, so that will give us the positive one and positive charge on the molecule. Okay, and this pKs we will not consider in this case because only one amino and carboxy terminal is there. So that will not make any change here. because the major group concerning about the charge of the polyal lysine is this amino group only. Okay, and yes, so this these endpoints we will not consider, and the rest of the amino and carboxy terminal are involved in making the peptide bond. So they will also be not considered. So at pH 7, poly D lysine will have a cationic charge on the molecule. Okay, so we'll move on to the next question. So polydilysine contains a hydrophobic sulfur group, hydrophilic sulfur group, hydrophobic amino group, or hydrophilic amino group. So as I discussed in the previous group also, so lysine has a side chain of amino terminal. Okay, and at pH 7, it will give a positive charge on the molecule. So it will like it is charged molecule, so it will be hydrophilic in nature. Okay, so hydrophilic means water loving. So everything that has charge will be water loving, and this is the amino group. So the correct answer would be it will have a hydrophilic amino group. Okay, so this is the correct answer. Now, moving on to the next question. So, polydilysine is used to coat substrate surface because of what charge it gives to the surface and what charge on the cell it binds to. So, as we were discussing before, also polydilysine has a positive charge on the surface. Okay, so as like this is considering this as the surface, and we are using polyl lysine as a substrate. So it will give a positive charge to the surface, which will help our cells that are negatively charged. Okay, so overall charge present on the cell is negative. It is maintained negative because of the potential that is maintained in the cell. So, uh, because of this, the cells are negatively charged and the lysine is positively charged. So they have a electrostatic interaction between them. So this is how polyl lysine helps in attaching the cells to the surface. So the correct answer would be polydilysine used to coat surface because it's positive charge. Okay, that binds to the negatively charged species on the cell surface. Okay, so the correct answer will be option A. Okay, so let's move on to the next question. In Alzheimer's disease, so it's a disease where this is a neurodegenerative disorder. So what will happen when the age proceeds? So the certain neurons present in our brain starts to die out. Okay, so this leads to people cannot remember their own name also, and this depends on many factors, being it genetic or being it environmental. So environmental being the age and how like the conditions that we are living in, being in stress, being what we eat. So all these conditions depend, gives the disease Alzheimer's disease. So in Alzheimer's disease, neurons of a specific region of the brain die up. So what region is this, and what type of organ it is present in? So in Alzheimer's disease, basically the hippocampal neurons die up. Okay. And it's present in the brain, not in the spinal cord. So the correct option is answer C. Okay, now let's move on to the next question. So which of these is not an enzyme? So what happens is generally when we name the enzyme, so we use the substrate name. This was the primitive way of naming an enzyme. So uh, like we use the substrate name in which this enzyme is acting, plus we have add ACE. Okay, so A, S, and E. So, for example, if there is a substrate called maltose and the enzyme that acts on it is maltase. Okay, so this is gives for the enzyme and this is the sugar that this enzyme is acting upon. Okay, this is the one form of naming an enzyme. All the glycolysis enzymes that we see are named in this respect only. Okay, and secondly, the other thing that the enzymes are named on the basis of substrate. that they are acting on plus the type of reaction. Okay, so in this case, like if ATP is involved, then kinase. If any NAD or FAD kind of molecules are involved, then they are named as dehydrogenase. So like that, the some enzymes are also. Okay, so this is the second way. Now the third way is from which organ or fruit or anything we are isolating. So this depends upon the scientists that they identify different enzymes from different sources. So like from any fruit, like for example, here we have papaya. So it is basically isolated from papaya. That's why its named is papaya. Okay, so likewise, we also named some of the enzymes. So these are different categories of naming of enzymes. Okay, so now here we'll consider the what are the different enzymes that we do. So papain is an enzyme. I just discussed about it. It's a protease. Okay, so protease is also an enzyme. It's a broad class of enzyme that acts on proteins. Collagenase, yes, this is the first category of naming of enzymes. So collagenase acts on collagen. Okay, and collagen is basically a carbohydrate. Okay, it's not an enzyme. So the correct answer of the question is option D. That is collagen because it's not an enzyme. Okay, now let's move on to the next question. In neurons, information or signal transfer along the neuron occurs electrically and transfer along synapse occur predominantly. So we have to specify if we see the structure of a neuron. Okay, so sorry. If we see this is the structure of a typical neuron. So this is the cell body that we see. These are the dendrites, and here is the axon, and these are the synaptic terminals of the axon. So first, the signal is received from this end. Okay, so these are the dendrites that receive the signal from different neurons to this particular neuron. Now, this signal will be received from here, and it will be sent to cell body. Now, this signal will be decided whether the signal needs to be sent further or not. So now from here, the signal will be sent from this way through the axon. So the signal that one signal when neuron receives from the dendrite transferred to the synaptic vesicle, synaptic cleft or the synapse part is through electrical means. Okay, in the form of an impulse kind of a thing. So what happens? There are certain gated channels that open receiving certain electrical responses. So they open and there is inflow of certain ions and outflow of certain ions. That changes the electric potential of that particular area. So in that like this, the electric potential changes and this is the movement of the impulse from dendrite to the synapse. Okay, so this happens in electrical way. That this is basically given in the question. Now, what if the signal needs to be sent to the other neuron? So consider this as the dendrite of the other neuron, and these are the synaptic vesicles, a synaptic cleft or the postsynaptic neuron. Okay, so now from here, if signal has to be transmitted to the other neuron. So what will be the case? Here, certain vesicles will be formed containing the neurotransmitter. So now, from here, the signal will be transmitted to other postsynaptic neurons using these chemicals. So now the signal is transmitted here chemically. Okay. So along the synapse, the transfer of signal occurs through chemical means. So the correct answer would be option B. Okay. So this is how the synapse occurs. And if we suppose if we have to if we are touching something, so we have to keep our hand there itself, or we have to remove our hand. So such conditions, in such case, the electric responses come from brain, and through this process, it transfers from different parts of the brain to the hand and vice versa. Okay, so let's move on to the next question. So which of these is not a part of hippocampus? So hippocampus basically consists of like there are many parts of hippocampus which are out of context in this scenario. So for the basic needs, we know that it has dentate gyrus, CA1. CAs have a region in the hippocampus and they are named ranging from CA1 to CA4. They have pyramidal neurons, and amygdala is a part of the limbic system. Okay, hippocampus is also part of the limbic system that controls basic emotions of our brain. So hippocampus, thalamus, and all amygdala, they constitute the part of the limbic system. So amygdala is not a part of the hippocampus. Okay, so this is the other part. So correct answer is amygdala. Now let's move on to the next question. So which of these is not a constituent of B27 supplement? So B27 supplement is used for culturing the neurons, basically. Okay, so basically when we are culturing neurons, we need a defined system. Okay, so defined system means we know everything about the media that we have. Okay, so every constituent is known. That's why it is called as a defined medium. What is undefined medium? Undefined medium is which we do not know what is the complete constituency of that particular medium. Okay. For example, there is a bacterial medium that we use that is a minimal medium or the agar, luria agar or something like that. Okay. So luria agar basically consists of trypton broth or yeast extract. Okay. So we say that it's a source of nitrogen, but we do not know the exact composition of yeast extract or the trypton broth. So that's why it is uh, plays in the category of undefined meat. So here, B27 supplement is used for culturing the neurons that comes under the defined medium category. Okay, so why B27 is called here? Because 27 supplements are being added into this. Okay, so what are the supplements from the given options are not present in this? So poly-D-lysine is basically a substrate. Okay, it has never been added to any media. So this is not present in this particular any media, not being B27, it is not present in any media. Okay, so BSA is there, catalase is there, and human recombinant insulin is there. Okay, so yes, correct option is A. And this is the basic. So this is the constituency of the B27 medium. So it has basic vitamins, biotin, alpha tocopherol acetate, alpha tocopherol vitamin A. It has certain proteins also, BSA, fatty acid prefraction, catalase, human recombinant insulin, human transferrin superoxide dismutase, and it has other components as also. For example, this is a hormone, cortisone. Sorry, so it is a hormone cortisone, it is sugar, galactose, ethanolamine, glutathione, and carnitine, linoleic acid, progesterone, and so on and so forth. So these new components we know that it is being added to the B27 medium. Okay, that's why we know that it's a defined system, and these components are useful in growing neurons in cell culture scenario. Okay, so This is the condition. Now let's move on to the next question. So Leibovitz L15 media contain this but lacks this particular compound. Okay. So basically L15 media is used for so so L15 media is used in the case of growing CO2 free cell culture. Okay, so how can we do it? We have different buffering compounds uh, that helps in buffering of the cell culture system. Okay, so that we can have we cannot use CO2 cylinders for growing the cells. So here we add sodium bicarbonate, glutathione, and all those things that helps in the buffering action inside the medium. So the basic component is sodium bicarbonate that provides the CO2 or the carbonic acid that we need for buffering action mainly. And other components are also there that helps in buffering of the media. Okay, so in this case, it also has sodium pyruvate for the same purpose as well as for metabolizing. So L15 media has sodium pyruvate, but it doesn't have sodium carbonate. It has sodium bicarbonate. Okay, so the correct option is option A. Uh, Sorry, the answer is written wrong here. It must be option A. Okay, because it will have sodium pyruvate, but it will not have sodium carbonate. It will have sodium bicarbonate. Okay, now let's move on to the next question. So P1 to P14 rat is a good source of. So actually P, it must not be P, it will be E. Okay, E means that it's an embryonic condition inside the rat. So when like mating occurs and the zygote is formed, from that case, it's considered as a embryonic condition inside the womb of the mother. So from day zero to like for in case of rat, the parturition happens around 24 days. So from day zero to day 24, it is E. And after 24, when the parturition happens, so this is the birth. So what will happen here? Here it will after from here it will be considered as postnatal. That is after birth. So from here it will be P1 to the extent we are counting it. Okay. So this is the thing. So from E1 to E14, rat is a good source of dash to study myelination of PNS. So here the correct answer is Schwann cells. So what Schwann cells basically do, first we'll tell the position. So considering this as the cyton, so here the dendrites are there, and this as the axon of the neuron. So what will happen? There are certain sheath-like structure. Okay, so these are the myelin sheaths that we see. And here we have certain cells. These are called as Schwann cells. These cells helps in secretion of these kind of myelin sheaths. So this is basically a lipid sheaths that helps in uh, uh, saving the neurons with the electric shock. Okay. So Schwann cells helps in secretion of these myelin sheaths. So they helps in myelination. So when we want to study the myelination of PNS, PNS means peripheral nervous system. So anything outside the central nervous system will be comprised as different organ. So when we are dealing with anything other than the central nervous system, so in that case, Schwann cells will be isolated from these particular neurons. Okay. So then only we can study the myelination of different organs. Oligodendrocytes are also used for myelination, but this is the case of CNS. Okay, so in case of CNS, the myelination is studied with the help of oligodendrocytes. Okay, so the correct answer is option C. Now let's move on to the next question. So why E17? That we just talked about the naming of this particular number name that at particular point we isolate certain organs. So is a perfect time in mice development that is used to isolate hippocampus from the rat tail. So the options are: it is a good source of pyramidal neurons. Hippocampus is fully developed at this point. Neurons are fully differentiated at this point, or more than one option is correct. So what will happen at E17? So what generally happens is, hippocampus is generally a good source of pyramidal neurons. And it at E17, it is fully developed inside the brain. So if we have to culture this particular organ and isolate neurons from this, so now we know that it is fully developed and it doesn't, it can uh, do its function on its own. So now we can isolate at it and grow in culture and study different aspects that we want to study. For example, like in the lecture, it was discussed about the Alzheimer's disease. So that's how it is cultured in the in vitro conditions. So, like the correct answer would be option A and option B. So, more than one option is correct. So, option D would be the correct answer. Okay, option D is correct. Now, move on to the next question. So, best way to isolate hippocampus neurons is, so we can use, like, if we have to dissociate any neuron from any organ of the body. Okay, so for example, consider this as the brain, and here we have the hippocampus neuron. So if we use an enzymatic method, so that has certain advantages as well as disadvantages. So in case of advantages, what we see, we have ECM, that is the extracellular matrix. So these particular enzymes helps in removal of the ECM. Okay, but what happens is when we use the dose of this enzyme, so generally what happens is there is some toxicity related to these enzymes, and these enzymes are basically proteases. So they cause very much like harm to the nearby cells. So what will happen? The brain cells that we are isolating can also be damaged. So in this case, what we do is we generally not prefer to use enzymes here. Okay, so this will be the disadvantage that the enzyme, the neurons are also harmed by using these enzymes. So like enzymes are generally not preferred to use, but we can use. It's not the case we cannot use, but we have to optimize the time for which the enzymes are being used. Okay, so we use a mechanical method that is mostly preferred in this case. Okay, so it depends upon the lab we are working in and what are the conditions that we are using to isolate the organs or the neuron. Okay, so the mechanical method is basically called as trituration. Okay, so this is the mechanical method that we are talking about. So, like the in current scenario, it will be context dependent or lab dependent, but mechanical method has advantages over the enzymatic methods that we are using. So the correct answer would be trituring as compared to protease digestion. Okay, but yes, papain digestion is also used in many labs for isolating hippocampus neurons, and trypsin digestion is generally not used for hippocampus neurons. It is generally used for isolating neurons from the cortex region of the brain. Okay, so the correct answer from our case is trituring. Okay, so let's move on to the next question. So you have just seeded certain hippocampus neurons in a cell culture plate. After what time? you will change the first media. So, sorry, this is the technical question. Okay, so when you have certain experience in cell culture, so what we generally do is if we have certain primary culture, so we are using adherent culture system. So we have a cell culture plate. We have added all the cells from the media, being it primary culture, or when we are reviving the cells also from liquid nitrogen. So what we do is we add this, we add the media, and we keep it for at least 1 day, 24 hours. And then we see at that point, we change the media. And what is this time point? This is the time point needed for the cells to fix to the surface. Okay, so this time is taken by the cells for fixing to the surface. That's why we take 1 day, and after that point of time, we change the media. And there is another reason is that when we are reviving cells, so they take up more amount of media, and the media that we use for reviving the cells is different from the media that we use for normal maintenance or passaging of cells. Okay, so this media would have high amount of serum that we use. Okay, though, so in this media that is used for reviving or the first culturing of cells, the serum is high in this condition. So we have to change the media after 1 day because now the cells will have toxicity of serum. Okay, so this can be in many ways that we'll discuss in the next week. So like this is why we have to change the media after 1 day, but we cannot wait till this time that the cells has been fixed in the on the substrate or not. Why? Because if some of the cells have not been fixed on the surface, they are floating in the medium. That means that they are dead. Okay, so this is the second reason that we have to remove those cells that are dead from the media so they do not release any toxic compounds in the medium affecting other cells also. So we change the media after this time and we do not wait for the cells to stick to the plate. Okay. So we see the plate if the cells are stick or non-stick. We change the media after 1 day. Okay. So this is the scenario. So the correct answer would be after 1 day. Okay. So let's move on to the next question. So what will act as a control in any drug reactions using cell culture system? So like when we use any drug system or anything we are testing, okay. So we have a control system. So we have two groups, control or test groups. Test groups can be anything, being different concentration of drugs you are testing, or you have different compounds to test. So these comes under the test group. Now, test group will be made with some base solution. So this is not that acid-base wala solution. This is the solution in which your drug would be dissolved. Okay. So generally we use water or any other buffering reagent, for example, PBS. So we use any of these things for dissolving any drug solution or any enzyme solution that we are testing. So this will act as a base solution that we use. Now, when we treat any cell with any of these drugs, so they have dual effect. First of the drug and second of the base solution. Okay. So now when we add any drug to the media, the effect of the drug is solely or it is from the solution that it is dissolved in also. So we have to test this thing also. And what will happen in this case? So generally this base solution is non-toxic to the cell, but we have to consider the fact that it can be toxic to the cell in any cell. So the test group will have both the things and the control group will have only the base solution. So that can be water, PBS, or anything. Okay, so in this case, we have the base solution, and that will act as our control in every case. Okay, being we are testing the absorbance or anything. So this is why we use control groups in our experiments. And that will help us to compare the different results that we are obtaining from the test group. So the control group are compared with the test group, and significance is found in that scenario itself. Okay, so the control that we use in any drug reaction using cell culture system is the solution used to make drugs added to the cells. Okay, so this is the correct answer, option A. Okay, so let's move on to the next question. You are studying the dendrite formation in hippocampal neuron. Which protein would you stain? So as we know, like we have discussed in previous questions also. So when this consider this as the axon of the neuron, and here it will form certain branches, and these are the synaptic clefts. Okay. Now at this point of time, like this in this area, it will have some bulbs. Okay. So these are called as synaptic cleft or synaptic button. So now if we have to, there will be certain proteins that will be only present in this particular case. Okay. So that will be that will help us to see what like the synapse is present in a particular neuron or not, or it is developing or not. Similarly, when we see this is the cell body, these are the dendrites. There will be some different proteins present in the dendrites. Okay, so that will be different from the synapse. That will help us to identify the dendrite formation. So here the correct answer is option C. So MAP2 basically helps in formation of the dendrites. So it will help us whether the dendrite formation is happening in the hippocampal neuron or not. So when we are studying different drug reactions or different development patterns of the neuron in vitro, so we use MAP2 protein, and that will help us to allow whether the dendrite formation is taking place or not, and how the dendrites are being formed. So we use certain visual markers also, like we can bind MAP2 with EGFP, and in that case, what will happen? And transfect these neurons with that particular EGFP bound protein. And here we'll see green dots in that case. Okay, like this. We'll be seeing green dots here in the dendrites formation. And if the dendrite is growing, then that green dots will also grow like this. So when initially the dendrite was this size, and then it grew to this size. So this is how we see the dendrite formation or dendrite increase in length using macro. Okay. So there are different proteins that are used. So synapsin is used for testing the synapse. Okay. And chirin G is used for studying the axon. And for cell body, we use a tubulin protein, tubulin and F actin. Okay. So these are the different proteins that we stain the neurons, and we study different uh, formation of different parts of the neuron. Okay. So now let's move on to the next question. So you fuse synaptophysin with GFP and visualize the neurons under a microscope. Found that there are 13 green spots are present after a long tube structure like structure. What can you conclude from the observation? So, like synaptophysin is also present in the synapse, and it is being used to visualize the synapse structure. So, of 30 spots that we see. So in the structure, it has been mentioned that after the long tube-like structure, at the end of the neurons, there is a particular 30 green dots are being observed like this. Okay. So what we see, we have these synapse in this condition. Okay, like this, it will be seen. So it will be 30 synapses are present there. Fine. So synaptophysin is basically used to like visualize the synapse using GFP, and here we can see for example, if we are testing a drug for Alzheimer's disease, so we can see whether the drug is working or not, the synapse is forming or not, it is making link with other neurons or not. So in that case, it will be the so in this question that we see, the green spot basically represents synapses. Okay, so option A is correct. So now let's move on to the next question. Why is glutamine not added directly to the cell culture? So what happens is in case of a cell culture scenario, what we are seeing here. So what will happen here? The if we add glutamine to the structure, it degrades simultaneously when we add in this medium due to different components of the medium as well as due to the cells present in the medium. Okay, and it will be toxic because the end product that is formed after degradation, two products are formed. First is ammonia, and other is uh, pyrrolidine carboxylic acid. So these two products are formed, and these two are toxic for the cells. Okay, so uh, these are not used in this case. So it degrades in the media and releases toxic ammonia, and pyrrolidine carboxylic acid. That's why the glutamine is not directly added to the cell culture. So option D is correct. So now what is added as a substitute for glutamine? So glutamax is added as a substitute for glutamine, and it is this? It's a dipeptide for glutamine. So Q, Q. So why I have written Q here? Because it's the single digit denomination for glutamine. So it's a dipeptide. So Q and Q. So it is basically glutamax, and it like when we add it in a media, it releases glutamine that is used by the cell. Now, what is glutamine used for? Used for protein formation as a source of nitrogen. Okay, so this is the basic use of glutamine as glutamax. Okay, now let's move on to the next question. So you have three drugs to test for treatment of Alzheimer's disease. AD is a short form of Alzheimer's disease. And you found that the number of synapse buttons are higher in control group as compared to the treated. What can be the possible inference of the above experiment? So if we depict this particular question in form of a graph, so here are the number of synapse or synaptic buttons. Okay, and here we present group 2. So we will have four groups here. First will be the control. Next will be the drug 1, drug 2, and drug 3. These four groups will be there. And here we chose some arbitrary numbers for the question sake. So we know that number of synapse buttons is, so suppose it's 7 in the control group. And in case of the D1, it's 5. In case of D2, it's 3. And in case of D3 is 2. Okay, these are arbitrary numbers, just I'm using for explanation of the question. So what will happen in this case? This 3 will be significantly down-regulated as compared to the control. But in case of Alzheimer's disease, if we have to use a drug, the control groups must have higher synapse number as compared to the drug test groups must have higher synapse number as compared to the control group. So what can be the reason? There might be the drug are causing some negative effect on the neurons, or they might be toxic to the neurons. Okay, that's why the neurons are dying when we apply those drugs. Or secondly, they can be apoptotic to the neurons. So when we add these drugs to the particular cell culture system, they are basically apoptotic side. They are going into degradation. Okay. So yes, the drugs have negative effect on the hippocampal neuron, or the drug concentration was toxic to the neuron. Okay, so these both can be the reason why the drugs are not showing good results in this case, and the control group has high number of synapses as compared to the drug control. Okay, because in Alzheimer's disease, that's a degenerative disease. So if we have to see whether a drug is working or not, we have to see the number of synapses increasing after treatment of the drug. So we see whether the drug is helping the neurons to form the synapse, or it is not helping. So here in this case, it is not helping, or it has a negative impact on the neuron itself. Okay, so this is the question. Now we move on to the next question. So yes, option D is correct. Now we move on to the next question. So this is a numerical based question. You have 4 lakh cells in a 5 ml cell suspension. You decide to place the cells in on a 15 millimeter of side length square slips. So for example, this is the cell suspension you have. This volume is 5 ml, and these are 4 lakh cells. Okay, now you have a square cover slip, which is 15 millimeter into 15 millimeter. So at density, you need 500 cells per millimeter square. Okay. So this is the scenario. So from this, how can we calculate what volume of this suspension can be added to this particular cover slip? Okay. So first we'll see how many cells do we need in this particular area of the cover slip. So the area of the cover slip will be, as it's a square cover slip, so this will be L square. So this is 50 into 50 millimeter square. Okay, now total number of cells to be added to the cover slip would be how much? 500 into 15 into 15. Okay, these many cells we need to add on this particular cover slip. Now, from these cells, for these cells, how much volume of this particular suspension needs to be added on this particular cover slip that we have to calculate. So this here we apply simple unitary method. So we have 4 lakh cells in 5 ml of suspension. Now, if we have to have 500 into 15 into 15, then how much volume we need? So this will be 5 divided by 4 lakh into 500 into 15 into 50. So these numbers, this will be in ml. Okay, so this amount of volume will have these number of cells, and that will be added to that particular cover slip. Okay, so this is in ml, but we have to find the amount in like microliters. So from the conversion, we know 1 ml is equal to 1000 microliters. Okay, so this you have to remember this is from the conversion that we use in basic unitary method. So we the answer would come around 1.4 ml. Okay, so this will be equal to 1400 microliters. So the correct answer would be option A. Okay, similar kind of questions can be asked in same scenario. So like you counted the cells in a hemocytometer that have four quadrants, and the number of cells can be n. What is the dilution factor you are using, and the conversion number divided by the number of quadrants you have counted. Okay, so from this formula, you can calculate the number of cells, and you would know how much volume of cells you have. From that scenario, you can calculate the density of cells to be added per millimeter square, like this. So the question can be framed in many ways, or else like there can be another question where you have the number of cells with you, and you know how much cells you have to add on a particular cover slip. And like for example, you have n number of cells with you, and you know small n number of cells you have to add on a cover slip. So how many cover slip can you add with those particular cells? So n divided by n will give you the answer. Okay, certainly the question can also be changed in this case that the cover slip that you are using are different shapes. For example, it can be rectangular, it can be circular, it can be triangular. So any scenario can be used, and you must know the area how which we calculate the area of these particular shapes. So in on this basis, different questions can be made. Okay, for calculations of particular cells to be added on certain amount of media or something like that. Okay, so this was the last question for today, and thank you for guys for your patience. So we'll be closing the session now. So I'm ending the meeting.